All right. So, this is the introduction. So, microbiology studies small things. How small? The smallest ones are, that we're going to talk about, are viruses. And usually to see them, you need an electron microscope. Now, is that true for all viruses? No, it isn't. The largest viruses, not even the smallpox, but viruses like Pandora virus, you can fairly clearly see in the light microscope. Does that make sense? So we can say that in general, you need an electron microscope to see viruses. But there are exceptions from the rule. For other microorganisms, such as bacteria, oh, for the love of God, bacteria or archaea or protozoa, okay, usually you can use light microscope and it's fine. Some bacteria are super small, so you better be using electron, but overall, that would follow the suit. So far, any questions? We're good? Moving on. So, in general, this is the sequence of sizes of microorganisms that we're talking about. Viruses are smallest, helminths are the largest. As a matter of fact, helminths most of them can be observed with the naked eye. Which brings us to the conversation about main structural features that characterize each individual group of microorganisms. So first of all, we can describe microorganisms as being pro or eukaryotic, okay? You may remember that eukaryotes, they have a nucleus and the membrane-bound organelles, while prokaryotes do not. Clear? No? Okay. Now, prokaryotes, without an exception, are unicellular. There are no multicellular prokaryotic organisms. Why? Well, it's a good question. Most likely because prokaryotes cannot establish sufficient communication between the cells. Now, the branch of microbiology that studies bacteria and archaea, those prokaryotes, is called bacteriology. And then we have a bunch of eukaryotic organisms. Algae, which can be unicellular, like this, Emiliana huxleyi, or multicellular, like kelp. And the branch of microbiology that studies them is called phycology. You should know that. Protozoa, their feature is that they are eukaryotic, which are always unicellular. There's a definition. And then, of course, we have fungi. Fungi can be unicellular, like yeast, or multicellular, like mold. And the branch of microbiology that studies them is called mycology. Finally, helminths. Those are the only organisms in that list that are always multicellular. Again, the eukaryotes. Now, what about these guys? What about viruses, right? So if you look at the viruses, like this filovirus, viruses do not have cell structure. They don't have cells, which means you can't really characterize them as being unicellular, as being pro or eukaryotic. All good so far? Clear? Now, you don't need to know the examples. It's a rule. So how do we classify microorganisms? Originally, the classification of living organisms, well, originally it was Carl Linnaeus, but Whittaker classification that was widely accepted mid-20th century relied on 5 kingdoms: Monera, protists, plants, fungi, and animals, which were based on the observable characteristics, such as structure or ecology. Now, I think you don't have any problem with identifying members of fungi or animals on plants, but what about Monera and protists? So Monera are all prokaryotes, and protists are single-celled eukaryotes. Now later, I believe in late 1970s, Carl Rose developed his own system based not on structural similarities, but on the genetic similarities between very conserved, very stable genes. And he demonstrated, along with his colleagues, that all organisms, based on their genetic relationships, evolutionary relationships, can be grouped into 3 domains: bacteria, archaea, and eukarya. If you look at this phylogenetic tree, effectively 23andMe of nature, you can see that this last universal common ancestor gave rise to bacteria, and another branch revealed archaea and eukarya. Paradoxically, eukaryotic organisms are closer to archaea than both of them are to bacteria. Effectively, archaea and eukaryotes are brother and sister, while bacteria is their cousin. We doing all right so far? Now, what about naming? We use binomial nomenclature for all living things, which effectively excludes viruses. When you look at the name of the species, such as Giardia lamblia, these 2 words represent, well, again, the name of the species. Within that name, the first word, Giardia, refers to the genus. It is applied to all living organisms from bacteria to humans. The first word represents broadly the genus, and both words name the species. Viruses have the most awesome names. Plant viruses in particular, this is the real virus, which the name describes what it specifically does to tomato plants. Okay? So I kind of cut on the history of microbiology, just FYI, microbiology. The idea that diseases can be transmittable appeared in ancient times among Greeks and then Romans, and Romans were the first civilization to build a proper sewage system, which greatly reduced the diseases associated with the gastrointestinal tract. Okay? But the ultimate question that biologists had to answer is whether life can appear from non-living things, so-called spontaneous generation. The first person to question that was, seriously question that, was Italian scientist Francesco Redi, who held meat in 3 containers. One container was open, so no doubt that there were some maggots. You know, flies were landing and laying eggs. Another container was tightly sealed. There were no maggots, which from our perspective makes perfect sense. There's no contact. And then there was this. So there was an exchange, so to say, in terms of the gas from meat to outside world, but flies nevertheless couldn't land on meat. So even the gauze that prevented the contact of living things with non-living meat demonstrated that non-living objects probably cannot give rise to life. And then there was famous Pasteur experiment here, in which he compared two flasks with curved necks. These flasks were effectively sealed by this water seal after being boiled. So this flask, he snapped the neck and exposed the content that was sterile to the environment, and eventually, in a few days, he saw the growth. To my knowledge, this flask, which neck remained intact, is still presented in Pasteur Institute in Paris. This experiment was done 130 years ago. That's pretty impressive. Okay? So that was pretty much the final blow into spontaneous generation theory. And then a bunch of Germans came up with an idea that actually the building block of any organism is a cell, and a cell can be derived only from another cell. So here we go to the modern biological concepts. Now the connection, the actual solid connection between the idea of disease being caused by infectious agents started in the mid-19th century with Ignaz Semmelweis, Hungarian by nationality, physician who worked as a chief medical officer, let's call him this way, of a hospital in Vienna, Austria. You noticed, so back in the days, a lot of women postpartum developed sepsis. It's called puerperal fever. You can see that in some years it was 1/3 of all women who developed sepsis. It's 19th century, there are no drugs. You can't really treat it. So those women were doomed. Semmelweis was not happy about that, and he noticed that women who were taken care of by midwives had better chances at survival. So he was wondering what's going on. And turns out there was one location in a hospital which was often visited by residents but not midwives. Anyone wants to venture a guess? Nope, not OR. More notorious.
The bathroom?
The morgue. Residents had to practice on cadavers. And they would practice, no gloves, folks. They would do the dissections and go back and deliver babies. Sounds incomprehensible by the modern standards. So Semmelweis simply introduced hand wash with chlorine, and the rate of sepsis dropped. Okay? That make sense? Later, same century, Joseph Lister, the British surgeon, introduced this interesting contraption that sprayed carbolic acid, effectively water solution of phenol, on the patient's skin and wounds during the surgery. Little detail, just FYI. It was the first antiseptic technique, or aseptic technique. Right? Make sense? There was no anesthesia back then yet. Yeah. Kind of sucked, right? Well, before it was worse. No anesthesia and no aseptic technique. So these were two pioneers in microbial control in a hospital. But at the end of the day, it was Robert Koch who proposed his famous Koch's postulates, allowing future researchers to establish the connection between a microorganism and the disease that it potentially causes. Koch himself discovered Mycobacterium tuberculosis. He discovered Vibrio cholerae, a bacterium that causes cholera. Something else. I don't remember the third one. Okay? Good? So obviously, microorganisms are important. At some point, I was trying to come up with the location on Earth which does not have any microorganisms, and I actually figured it out. Anyone wants to guess? Which places on Earth do not, sterile completely? So again? Can't hear it. No, absolutely not. No. It's not real, but there are microorganisms. Well, you get, like, seriously, dude, yeah, nothing, yeah. I mean, Earth, like the stuff that we can get to. Nope. There are many corpses there, by the way. Yeah. Volcanoes, something hot, not survivable, right? Cold is not a problem, the heat is. So microorganisms are everywhere, except volcanoes. I'm going to say the triviality. Microbes cause diseases. Right? Right? They do. I'm not going to go through this table, although it is very interesting since our time is somewhat limited. I just want you to understand that, especially in developing countries, infectious diseases is the #1 cause of mortality. Okay? If we take Africa, what do you think is the main killer? Malaria is about third, fourth. Huh? No. HIV. #1. Tuberculosis. Go ahead.
You're going to say it, right?
What?
Like the answer to the question.
Yeah, HIV is the #1 cause.
Oh.
What question?
Oh, I thought you were asking me.
I was asking, what do you think? There were proposals that it's malaria. Malaria is top 3.
I was going to say like something with E. coli.
Nope. So, infectious diseases of gastrointestinal tract in general is in top 5.
When you say infection disease, you mean the disease that caused by microbes? Like bacteria and
Viruses, fungi, not diabetes. Diabetes is not an infectious disease. Or cardiovascular event is not an infection. Does that make sense? So, they cause a lot of deaths. Okay? You can look at this table when you have time. It's kind of interesting, the dynamics in the U.S. Like we had the mortality from vaccine-preventable diseases in 1980 was 2.1 per 100,000. 2014, it's 0.8. Ideally, it should be 0, or very close to 0, but, you know. I'm looking forward to the current Department of Health and Human Services administration because it's going to be a very, very exemplary shit show in the next few years. Bacteria can be useful. For example, on this graph, you can see an observation of the wine microbiome changes during fermentation, which may allow people to track the perfect wine, to mature it the best way possible. This god-awful and very interesting chemical pathway here demonstrates that bacteria can be used to produce solvents or butanol, the kind of alcohol that can be used in cars instead of gas. It's still experimental. And bacteria, of course, since they have to compete with other bacteria, can produce antibiotics, chemicals that kill the competitors. Okay? All good, folks? Everybody? All right. Which brings us to the first group of pathogenic microorganisms. I peppered the course with a bunch of pathogens to make it a little more interesting. So these are Clostridia. All Clostridia have something in common. They are obligate anaerobes. What does that mean?
They cannot have oxygen.
Correct. They cannot survive in the presence of oxygen. And they form endospore, structures that protect their genetic material, making them very hard to control chemically or physically. Okay? All good? So 4 common Clostridia species that cause human disease, presented here. So Clostridium botulinum, unremarkable looking bacteria here, causes the disease known as botulism, which is a flaccid paralysis because of, interruption in nerve transmission. People cannot contract their muscles. All good, folks? No? All right. Clostridium tetani, on the other hand, causes spastic paralysis, known as tetanus or lockjaw, when all muscles are contracted. Okay? Now, for any infectious agent that you will see in the course, you need to know, what it causes, what kind of disease, how it is transmitted, and whether or not there's a vaccine. So, Clostridium tetani is the only one that can be prevented by vaccination. We're good? If you go into a nursing program in your clinical practice, your chances to see either of these two are very slim. Botulism is rare, and tetanus is rare too, thanks to vaccination, but who knows? Okay? Questions? Next goes, both of them are environmental. Clostridium botulinum, though, mostly is transmitted by eating food that is contaminated, but still environmental. Clostridium tetani and Clostridium perfringens, they come from the environment, from the soil, pretty much. Clostridium perfringens causes gas gangrene. That stuff, you'll see. Remember that these microorganisms are anaerobes. They want low oxygen environment. People with diabetes have a pretty bad perfusion, blood flow in their limbs, especially in the legs, feet, okay? So if there's a wound and Clostridia get into the wound, it's a perfect environment. That make sense? So you're going to see gangrene in people with diabetes. And finally, C. diff. I understand a lot of people here work in some kind of healthcare settings, so you're familiar with that scourge. It causes colitis, perforation of the large intestine. And 25% of people here in this class have Clostridium difficile in their guts. It's just a fact, it's a known fact. Now, why those people don't have C. diff? Because they also have healthy microbiome that prevents Clostridia from multiplying. Once a patient like that receives an aggressive antibiotic treatment, it wipes off the good bacteria. Clostridia flourishes and produces toxins that ultimately lead to that colitis. Good? Now, treating C. diff with antibiotics sounds kind of counterproductive. It's like extinguishing fire with gas. You probably can do that, but it's a risky strategy. So now the treatment of Clostridium difficile is focused mostly on the replacement of microbiome through fecal transplantation. Now, fecal transplants now are made from freeze-dried human feces. They're all tested, to my knowledge. I don't know if now, but a few years ago, Cleveland Clinic paid $40 per dump. They tested it, obviously, but you could get some money for the pool. Before that, it was enema, so fecal suspension from the donor was delivered through enema, which is, and in the very beginning, it was just people were drinking fecal suspension, which I know sounds disgusting, but it also defeats the purpose. Gut microorganisms usually do not survive your stomach, which is acidic. That's why they changed to enema, because it was a more direct way to populate. Now it's just pills. Okay? Sounds good? So Clostridium difficile is also environmental bacteria. You can find it in the soil, but it mostly resides in the guts of humans and other animals.
So treatment for C. diff is
Fecal transplant for the most part, yeah. Now we're going to jump back to a quick review. So here, water and pH. Things to know, things to remember. The idea of a hydrogen bond, the bond that is formed between partially negative oxygen in one water molecule and partially positive hydrogen in another. Okay, just know what a hydrogen bond is. They're very important in formation of DNA proteins. And understand the whole pH scale. Do not memorize this. 7's neutral, above 7's basic, below 7's acidic. Before, if you increase acidity, pH falls. If you increase alkalinity, pH rises. Good? Any questions? All right. Carbs. Things to understand. The composition. This is not a chemistry class. I am not asking you to remember any of these formulas. Okay? I will not ask you formulas. I will not ask you the composition of maltose or sucrose or lactose. I want you to understand at the basic level that we have a bunch of monosaccharides which you can combine together in a chain called polysaccharide. Another product of this reaction is water. This is why it's called dehydration synthesis. And if you want to break this chain back, you need just to add water and some enzymes. Okay? We good so far? So functionally, mostly energy and structural roles, it's just cell wall. Obviously, you can find carbohydrates and other molecules, but I'm not going to focus too much on that. Lipids, 3 types I want you to remember. First are triglycerides. Why do we call them triglycerides? Triglycerides consist of glycerol residue and 3 fatty acid residues. All lipids are nonpolar / hydrophobic. The use for triglycerides is mostly energy. So you need to know the composition, glycerol and fatty acids. When we get to phospholipids, we add one more component into the mix. Instead one of the fatty acids, you have a residue of phosphate. So it's phosphate, glycerol, and 2 fatty acid tails. Turns out that this part of the molecule, the part that consists of glycerol and phosphate, is hydrophilic or polar, while the tails that consist of fatty acids are hydrophobic, i.e. nonpolar. Any questions? What is the function of these guys? The function of these guys is to form a membrane, as we will see very, very soon. Okay? And finally, this interestingly looking polycyclic lipids, sterols on the left and hopanoids on the right. Their function is to reduce the viscosity of cell membrane. Cell membrane is not a solid structure, it's not a wall. It's basically very viscous liquid. If you add sterols or hopanoids, it becomes less rigid, more fluid. All right? So what's the difference between the two? Well, sterols, they regulate the membranes of eukaryotes. Hopanoids do the same stuff in bacteria. Any questions before we move to proteins? Sterols, they regulate the fluidity of the membrane in the eukaryotes, while hopanoids do the same function but in bacteria. Now let's get to the proteins. Functionally, proteins do stuff. 99% of cellular function is carried out by proteins. So the basic building block for proteins is an amino acid. If you look at amino acids on the right-hand side, you will see that they all have the same skeleton, the same part, and then they differ by their side chains, highlighted here in blue. So this difference in side chains as a very important consequence from the functional standpoint. If you take two amino acids and join them, they will be joined by the peptide bond in the reaction of dehydration. Are we good so far? You can keep adding amino acids, forming tri, tetra, pentapeptides, and eventually you will form a long chain of different amino acids. Make sense? This is called a polypeptide and represents the primary structure of the protein. Because amino acids are different and their parts interact with each other through hydrogen bonds, for example, the polypeptide chain will start folding. And it will fold into a secondary structure, two main elements of which are alpha helices, where amino acids, the polypeptide is folded, well, as a helix, or beta sheets, where polypeptide is effectively folded like a sheet, like that. Now, the interactions between the side chains of amino acids will determine the tertiary structure of the protein, the globule. So far making sense? For many proteins, that's going to be it. Many proteins represent just one polypeptide. They're usually relatively small. But a lot of protein special enzymes in the cell have multiple polypeptide non-chemically merged together. They do not form specifically chemical bonds, they interact because of the charges and so on and so forth. Okay? This assembly of several polypeptides is called quaternary structure of a protein. You must know what represents each structure and what are the main elements like alpha helix and beta sheet. So far, good? We effectively established that protein structure is defined by the side chains of amino acids. Do you agree with me? Interaction between side chains will define that. You know, you have two polar side chains, they may interact, two non-polar, they may interact, just like that. So if you change an amino acid and change the side chain, you can potentially alter the protein structure. And if you alter the structure, what else do you alter? The F word. You alter the function. The whole concept of mutations and subsequent amino acid substitutions, that's, there you go, that's, changes in a polypeptide. Another thing, a lot of those interactions that we observe within proteins depend on the chemical composition or the temperature. It means that the structure can be disrupted. Protein structure can be disrupted. We call it protein denaturation. Two glaring examples, the egg drop soup, Chinese cuisine, which is very acidic, and when the egg is going there, it denatures, the proteins denature. And second example is when people try to make the hair from curly to straight and vice versa. It's done with temperature and chemicals. Does that make sense? And it's not permanent because the proteins in the hair will eventually assemble its initial shape. Okay? Finally, nucleic acids. So these guys consist of nucleotides, 4 types, A, G, T, and C for DNA. For RNA, T will be replaced by U. Please do not focus on that now. It's Unit 2. For our intents and purposes, DNA is double-stranded, RNA is single-stranded. The fundamental feature here is that the strands in DNA, for instance, they are complementary. Which means if you have A in one strand, there's going to be T across it in another. If you have G in one strand, there's going to be C across it. Okay? So that allows coding. That allows the concept of a message, and what's important, this message can be copied and passed down from generation to generation. And this brings us to 2 most important concepts in microbiology: gene expression and evolution. So, when we are talking about the genome of an organism, we talk about the information about its traits, right? So like bacteria can have a gene for antibiotic resistance. This is a phenotypical trait. Bacteria can have a set of genes that will result in the production of capsule. Capsule is a phenotype. Am I making sense? So phenotype is defined by genotype. But I want to be a little bit more specific. So let's say we talk about a bacteria that everybody knows, E. coli. It has 5,000 genes. Each gene encoding particular trait. We're good? Do you think that all those genes are expressed, all those genes generating proteins at the same time? Probably not. So, gene expression responds to changes in the environment, what authors attempted to represent by this flowchart. But I think I came up with a much better analogy. It's an analogy of cooking. So imagine that over generations in your family, you accumulated 5,000 recipes. They stored in a book. This book is in your living room. It's your cherished possession. It's effectively invaluable because it's your family heritage, right? That's your DNA. Okay? All the recipes. And tomorrow on Friday night, your friends are going to visit you, and your friends is a pretty interesting and varying bunch. Okay? You have a person with celiac disease, so gluten is out of the question, right? You have a person with lactose intolerance, no lactose. You have a Muslim person, so everything should be halal, right? You should exclude certain meats. You have a guy who's Hindu, so other dietary restrictions. And then you have a vegetarian friend. So you are facing certain environmental challenges. Are you going to cook all 5,000 dishes? No. You're going to choose certain, what fits best. Okay? And in response to this challenge, the changing environment, you're going to start making a meal. Do you follow my train of thought? So you decided on the recipes. Now you need to bring those recipes in the kitchen. Remember, 5,000 recipes. It's a pretty massive cookbook. And it's super valuable. Would you take it to the kitchen? Like the book itself? What would you do then, if not?
Take a copy.
Yeah, but how do you transfer them?
You just write it down.
Write it down. Well, come on. It's old people's talk. You're going to take a picture with your smartphone, right? And take a picture, take it to the kitchen. But taking a picture is transcription. Okay? Well, I'm going to put picture here. That's a picture of the recipe. So you effectively have the same, you have text in the book, you have text in your smartphone, it's both texts, nothing really changed. Then you start following the instructions, and you are cooking, making a meal. That's pouring. You convert the instructions of the recipe into the product of an actual edible meal. Your friends aren't going to appreciate if they will come in and say, well, I have pictures on my phone, look, we can eat them. No, you can't. So that's the idea. You have to get all the way to proteins, for the most part. Does that make sense? Why I like this analogy? Well, first of all, because it's mine. Second, because it works really, really well. If you start thinking about the fate of RNA in the cell, those images on your smartphone, those recipes, are you going to keep them forever? Same is true for RNA. RNA degrades pretty quickly. Or let's say you copying by hand and you make a mistake. Okay? Is it a catastrophe? I mean, not really. You're going to screw up one meal, you're going to cook it again. If you put a mistake, if you put change in the cookbook, that's a different story. Okay, you can give a copy of the cookbook to your kids. It's passing DNA down the generation. Does that make sense? Awesome. Second concept is evolution. There's Dobrzański, who was a Polish biologist, evolutionary biologist, immigrated to the United States, I think in the 1930s when Germans started working in Europe. He said nothing in nature makes sense other than the light of evolution. So if you start asking yourself, why does that happen? Why is that microorganism, why does it look like that? The universal answer is evolution. The only problem, we don't know the history. Does that make sense? But I'm not even going to focus on antibiotic resistance when we talk about evolution. I'm going to focus on something else, the pandemic that we had recently. At the beginning of the pandemic, several pharmaceutical companies released antibodies, drugs that effectively reduced the ability of coronavirus to infect humans and reduced mortality, especially in elderly patients. Okay? So guess what happened in about 6-8 months? It's selected for the individual viruses that could avoid those antibodies. It was inevitable. Everybody understood that it's not going to work forever. People are buying time. But that shows you, like, since then, those original antibodies, if you will try to use them now, they have no clinical effect. It's been demonstrated. Virus evolution is random, but it's driven by selective pressure. So when you look at something that happens in cells, you have to ask yourself, what kind of selective pressure are we dealing with now? All good? So now we're going to get into the, grind of it. Morphology. I'm not going to read this table to you, okay? I can tell you what kind of question you can expect. What is the term for a curved rod? Vibrio. You will effectively have to match the term and the description. Does that make sense to everybody? So I'm not going to spend time on this. Obviously, some genera of bacteria named just because of the shape, like Vibrio bacillus. Now, how do cells maintain their cell structure? Well, here we're talking about osmosis. So first of all, what is osmosis? Osmosis is a diffusion of water towards the higher solute concentrations. Notice that we're not talking about the concentration of any specific solute. We're talking about the total concentration of everything, which is called osmolality, which depends on the number of solute particles, but not the exact chemical nature. Now, if osmolarity inside the cell and outside the cell, if it's equal, we call the environment around the cell isotonic. You probably remember that term from previous biology classes or chemistry classes. If the environment has a greater concentration of solutes than the cell, we call such environment hypertonic, and what happens is water leaves the cell, forcing it to shrink and often undergo osmolysis. Finally, if the cell has more solutes inside than the solution outside, we call such environment hypotonic, and in this environment water instead moves into the cell, resulting in the cell swelling. In case of many bacterial and fungal cells, it's not such a huge problem because they have cell walls which prevent bacteria or fungi from exploding. But what about protozoa that do not have, don't have it here, but I'll just talk about it. What about protozoa that do not have a cell wall? They usually live in hypotonic environments. So now I want you to, for a second, imagine that you are a tiny protozoan cell, just one. No cell wall, just a membrane. Can you imagine that? And the water constantly comes into your cytoplasm. It flows into the cell. You're supposed to explode, and yet you don't. What do you do to not explode? You can't. You have nothing to absorb. See again? Yeah. It's like a sump pump in the basement. Does that make sense? Protozoa constantly pump water out. Water that comes in. This is why if you look at protozoan cells, they're constantly moving. They're always eating. They're always in the hunt because they need so much energy. Good? Another question that I want to ask is this. You're probably familiar with a body of water, or rather brine, called the Dead Sea in Israel. It's extremely salty. And yet, there are bacteria and archaea that live in these salty waters. How do you think they manage to survive? Let me guide you through it. Go ahead. Yeah, but how? Of course, they have adopted, but how?
They somehow ignore the osmolarity.
You can't ignore the osmolarity. It's like ignoring gravity. You can do it, but just once. Okay? Huh? I mean, that is the answer that covers everything.
They just transaction.
So again?
They just do some transaction between these molecules and make some.
When we talked about hypertonic and hypotonic, did I ever mention a single number? No, I didn't. Because it's a relative parameter, right? Hypertonic environment simply means that there's more solids outside than inside, but no numbers. Now, cells would feel totally comfortable in isotonic environments. Would you agree? So those cells that live in Dead Sea effectively have the same concentration of solutes inside that's outside. They don't have to be the same solutes, though, but they maintain the same osmolality within the cell. If you will try to put those cells in like lake, they'll explode immediately. Even cell wall is not going to save them. Now, we're going to talk about the structures found in prokaryotic cells. Every prokaryotic cell has these four structures. I want to urge you from when we use the term, it is the exact term. Like nucleoid isn't nucleus, although they sound pretty similar. So all prokaryotic cells have nucleoid. Remember, there's no nucleus. So nucleoid is just the area where DNA is. Okay? They have cytoplasm. That fills up the cell. They have a membrane that surrounds the cell, bounds it. And they have ribosomes, which are small non-membrane bound organelles consisting of RNA and proteins, which translate mRNA, synthesizing proteins. Okay, everything else, I think we were kind of talking about the membrane, so just remember in regards to the cell membrane that it consists in almost all cells of the phospholipids in which some proteins are embedded. And that makes the inside, that part of the membrane, hydrophobic, and the surfaces are hydrophilic. Compounds can cross the membrane either by passive or active transport. We also cause passive transport diffusion, in which chemicals move along the concentration gradient from their high to their low concentration. In case of simple diffusion, the chemicals are nonpolar and hydrophobic, and they can cross the membrane directly. While in case of facilitated diffusion, these chemicals are hydrophilic, polar molecules, and this is why they require either channels or carriers. Now, if you want the diffusion, the movement along down the concentration gradient does not require energy. Active transport, though, does. In case of active transport, molecules are transported, for the most part, against the concentration gradient. Usually from their low to their high concentration. Okay? Active transport always requires energy and always requires transmembrane proteins. Now, we are getting into the parts of the cells that are optional. Inclusions, for example, shown here, are basically accumulation of chemicals within prokaryotic cells that serve different purposes, from nutrition to orientation in space. Plasmids, which pictures I don't have here, are small circular double-stranded DNA molecules that can carry beneficial genes. For example, genes for antibiotic resistance. Plasmids are not essential for the cell survival. And then we have endospores. Endospores are a very sturdy structure shown here that are formed under harsh environmental conditions. During sporulation, the formation of endospore, genetic material of a cell is protected by multiple layers from any pretty much environmental challenge. Okay? And endospores can survive not just for years, not for centuries, but for thousands and millions of years. When endospore ends up in the environment, that is good, food, water, endospore germinates, forming a vegetative cell. So sporulation is the formation of endospore, germination is the emergence of vegetative cell from an endospore. This is clinically relevant because of a lot of human pathogens. They, form endospores. Okay? Sounds good? Most bacteria have cell walls. So cell wall is located outside of the membrane, as you can see here and here. And it consists of long carbohydrate chains, so-called glycan chains. So glycan chains are represented here. And these glycan chains are connected by peptide bridges. Remember, peptides consist of amino acids. This is why we call this structure peptidoglycan. Peptido and glycan. So this peptidoglycan cell wall is different in different types of microorganisms. This is why we call some bacteria gram-positive and some bacteria gram-negative. Gram-positive bacteria have a thick layer of peptidoglycan just outside of the membrane. So their cellular envelope effectively consists of two layers, a membrane and a wall. Gram-negative microorganisms have a fairly thin cell wall right outside the membrane, and then outside of that cell wall there is another so-called outer membrane, which contains several important molecules such as lipopolysaccharides, molecules that can cause disease in humans, porins, molecules that facilitate transport. Now, the wall of gram-positive microorganisms also contains important molecules such as teichoic acid, which contributes to the adhesion of these cells to the surface. Overall, if you will compare gram-positive and gram-negative cell membranes, you can see the difference. Gram-positive has 2 layers and gram-negative has 3. Gram-positive contains teichoic acid, while gram-negative membranes contain lipopolysaccharides. Okay? Our attention to cell wall is not accidental, because a lot of antibiotics, such as penicillins, cephalosporins, bacitracin, vancomycin, they target the synthesis of the cell wall. Now, especially in gram-negative bacteria, this molecule called peptidoglycan, polysaccharide, lipopolysaccharide here, which consists of several sugar chains, carbohydrate chains, and fatty acid molecules, can trigger a very powerful immune response, very strong inflammation, okay, causing non-specific generalized symptoms. Not all bacteria have cell walls. A group of molecules, such as mycoplasma, lack the cell wall entirely. And not all bacteria have either gram-positive or gram-negative cell walls. If you will compare the cell wall of bacteria such as Mycobacterium tuberculosis, shown here, they do look like gram-positives with a thick peptidoglycan cell wall, but on the outside they have a layer of a pretty waxy compound called mycolic acid. It's effectively wax. Okay? One of the common things to mix up, bacteria that contain mycolic acid, mycobacteria, and mycoplasma, cells that do not have cell wall at all. Outside of the cell wall, some bacteria have a layer of carbohydrates called glycocalyx. You need to know 2 types. One type is capsule. It's a pretty sturdy layer that facilitates the adhesion of bacteria to the surfaces, making them more dangerous, and also protects bacteria from phagocytosis by your own immune cells. A more loose version of glycocalyx is a slime layer. It doesn't protect bacteria from phagocytes, but it does increase the adhesion, facilitating the formation of biofilms on the surfaces. Biofilms, most bacteria exist in the form of biofilms, such as bacteria on the surface of your teeth, walls of your gut, as well as bacterial biofilms can be formed on the ventilation tubes and catheters in the hospital, presenting a very major hospital-associated infection threat. Many bacteria can move, not all of them. They can do it using a flagellum. You don't need to know the exact details of the flagellar structure between, in a gram-positive versus gram-negative microorganism. You need to understand this, and it's very simple. The way flagellum operates is basically you have a rod with a hook, and at the end you have the filament. And when rod turns, the filament works as a propeller. That make sense? That's it. Bacteria can have one flagellum. In this case, we call them monotrichous. Two flagella on the opposite ends of the cell. We call them amphitrichous. Lophotrichous bacteria have multiple flagella, one side and peri. Trichous bacteria have flagella all over the surface. In addition to flagella, another external appendage, which unfortunately or fortunately does not facilitate the movement but increases the adhesion to the surface, is fimbriae, a bunch of hair-like structures on the surface that is sticking. And the third appendage that I wanted to mention is pili. Pili are long, tubular structures that can either facilitate adhesion or enable bacteria to exchange DNA, so-called horizontal gene transfer. These are called sex pili, and they are important because can increase the spread of antibiotic resistance. Now, go ahead.
Are the fimbriae and the pili, are those types of flagella?
No, that's a good question. No. Those are three absolutely independent, structurally different appendages. Flagella move, fimbriae adhere, pili adhere or exchange genes. Now, when bacteria move using a flagellum, then can either run or tumble. When they run, they move into a certain direction. When they tumble, they orient themselves according to the stimuli. What can be the stimulus to movement? It can be chemical, light, or even magnetic field. Now, what do we call movement? We call movement taxis. So if a movement is driven by chemicals, we call it a chemotaxis, right? Light, phototaxis. Bacteria can move towards something or away. If bacteria move towards a chemical, like a nutrient, we call it positive chemotaxis. If bacteria try to avoid light, that would be negative phototaxis. Okay, we're good? Now, if you start looking at the immediate source of energy for flagellar movement, you will be probably surprised that it's not directly ATP. It's a concentration gradient of hydrogen ions across the membrane. I'm pretty sure you're asking yourself, why is that relevant? Why is that important? Well, hydrogen ions are effectively acid, effectively pH. If bacteria ends up in the environment with suboptimal pH, it may lose the ability to move because of disturbed concentration gradient. But it's a movement of hydrogen ions, a diffusion of hydrogen ions, very much like in the oxidative phosphorylation, that facilitates the movement. So, probably the poster child of bacterial infections is Staphylococcus. So we're going to talk about 3 species. I'm going to knock out Staphylococcus epidermidis, which is a normal microorganism present on human skin. It's not really a pathogen. Staphylococcus saprophyticus is a very narrow area of pathogenicity. It's found in the human gut and transmitted mostly sexually during anal sex. And when it infects, especially men, male reproductive and male urinary systems are kind of merged at some point. So that may result in UTI, urinary tract infection. Men can pass it to women, and then it's urinary infection in them. The most notorious, the most well-known is Staph aureus. So it causes a variety of infections. Here is the illustration of what it can do on the skin. It can start with a fairly mild superficial skin infection known as impetigo, which is contact transmitted, and eventually develop into so-called scalded skin syndrome because bacteria start breaking down the connection between the epidermis and dermis, and skin is just falling off. If it infects hair follicles, it can result in furuncles and carbuncles. This is a furuncle. This is a legitimate carbuncle. Okay? When it starts crawling subcutaneously, it can cause cellulitis, but the most notorious, of course, is this. This is the necrotizing fasciitis, flesh-eating disease. Okay? It is treated by surgical removal or removal using sterile maggots, debridement, basically, antibiotics, very few times. As I mentioned, for all microorganisms that we mention in the course, there are some fundamental things that you must know just by default. Disease, transmission route, vaccine. So, transmission route for Staph aureus, either contact or respiratory for infections like sinusitis, not UTI, obviously. Sinusitis. And it also can be transmitted through blood. Toxic shock syndrome is the result of hyperactivation of human immune system by one of the toxins of that microorganism, and it was discovered when a bunch of women got sepsis by using female hygienic products, namely tampons, that contained naturally derived cotton. And this cotton was contaminated with Staph aureus. It entered the bloodstream and they had sepsis and many of them died. You don't need to know virulence factors, don't worry. Okay? Now, eukaryotic cell. I'm not going to go into much details. Let me explain why. It's probably 10th time we talk about the parts of eukaryotic cell. I will 100% ask you to match the part and the function. The part and the function. Okay? Now remember I mentioned the importance of terminology. Right? Terminology is important. Nucleus, nucleolus, and nucleoid are 3 different things. Just to keep in mind. Okay? So, I'm going to get back to the quiz. Okay, plasma membrane. Similar ideas in bacteria. Okay? Phospholipid bilayer, cholesterol to regulate fluidity, transmembrane proteins as signaling proteins or transport molecules, peripheral proteins, usually interacting with integral proteins as well. Okay, one sec. Perfect. So that's all pretty straightforward. Now, what about the maintenance of the cell shape? What about the cytoskeleton? So cytoskeleton in eukaryotic cells consists of microtubules, for God's sakes, that are made out of tubulin. Okay. Microtubules are dynamic, which means they can be destroyed and rebuilt. They're important for intracellular transport, for movement of cells, and for the cell division. Microfilaments consist of the protein actin. They're also dynamic, meaning they can be rebuilt, can be destroyed, and participate in similar functions such as movement, cell division, and sometimes transport. Unlike these two, intermediate filaments that consist usually of the protein keratin are stable. They're destroyed, means cell collapses. They are like beams and studs in the house. So you need to know what these filaments are made of, right? And whether they're dynamic, stable, and what their function is. That brings us to mitochondria and chloroplasts. Well, first of all, this is indeed the powerhouse of the cell. Mitochondria used to be an independent aerobic bacterium that could perform aerobic respiration. They were acquired billions of years ago by first eukaryotes, and both parties benefited from that relationship. Eukaryotic cells found the source of ATP. Okay? Bacteria received protection and constant supply of nutrients. We're doing all right so far? Same story is for chloroplasts. Those were photosynthetic bacteria billions of years ago that got accepted by that first generation of eukaryotes. Okay? Why do we think it is that? Why do we think it was endosymbiosis? Both of them can undergo binary fission. When eukaryotic cells divide, mitochondria and chloroplasts also divide. They're not synthesized from scratch. You can replicate them even in an egg. They have DNA, which is very similar to bacterial DNA, and they have ribosomes, which are effectively bacterial ribosomes. All that evidence, albeit circumstantial, pretty much confirms that they are endosymbionts. Similar to bacterial capsules, some eukaryotic organisms have extracellular matrix, which effectively can form a capsule. For instance, pathogenic fungus, Cryptococcus neoformans, forms a capsule around the cell. Mostly protoglycans in the protein collagen. Eukaryotes also have flagella. They look the same as bacterial, but structurally they're different. Remember how I compared bacterial flagella to a propeller? This is an oar, a paddle. Instead of moving in a circular fashion, flagella of eukaryotes, they wave. Cilia are structurally similar to flagella, but they much shorter, more numerous, while serving the same purpose, movement. Either a microorganism or moving food into, the part of organism that actually eats it. Okay, sounds good? So let's chat about some of the pathogens. I think we're just going to finish week 1 and then pop in the lab and do the lab. It's not a lot of time. So first category of eukaryotes, protozoa. You must know this classification. The classification is based on the movements. Celiophora move using cilia, which is like, yeah. Amoeba move using pseudopodia. Mastigophora use flagella, while Apicomplexa can't move at all. Now, if you look at the third column in this table, only Apicomplexa group has no species that live independently. All of them are parasitic. And that is exactly because they can't move. If you can't move, you can't chase food. You have to have food around you. Now, I mentioned before, you don't need to know the examples. I just provide them to you. And in this example, just to show, if the color of the example is blue, it's not parasitic. If it's red, it's parasitic. What about this guy, for example? It means that it's non-parasitic, but can be. And that brings us to one of my favorite organisms, Igleria fowleri. It's an amoeba. These amoebae exist in soil and in stagnant, more or less stagnant waters, like ponds and lakes. When human kids swim in the water, they can snort some of the water, and bacteria, this amoeba can go through the roof of the nasal cavity, through the cribriform plate, and the ethmoid bone, straight into the brain, resulting in primary amoebic meningoencephalitis. It's a very quick disease, and I want you to understand, for amoebae, it's the dead end. They trapped. It is not a communicable disease. If somebody has amoebic meningoencephalitis, they do not pass it to anybody else. It is often misdiagnosed as meningitis, treated with antibiotics, which means people lose valuable time. In most cases result in death. Some cases result in survival because it is treatable if it's early, but it leaves a lasting neurological damage. It's not frequent. I don't remember which year I picked up that statistic, but 1962 to 2008. So we're talking 46 years, we have 111 cases, not a lot. Some states don't have these cases at all, just totally explainable. This is what I love about epidemiology. If you look at the graph, people who get infected the most, kids. When they get infected the most? Summer. Where? South, because it's super hot. And remember, the map shows the total number of cases. So there's no surprise that Texas has the greatest number of cases because there's so many people. And it shouldn't come as a surprise to you also that Florida is the second because, well, it's Florida. Okay? Now we jump to another eukaryote type fungi. Good news. None. The only classification that I want you to use, and I know that it's not a proper taxonomy, but it's useful. Difference between molds, yeasts, and dimorphic fungi. Molds are multicellular, although cells can fuse sometimes. They form hyphae, the fuzzy appearance, right? While yeast are unicellular. These chains of cells actually are chains of individual independent organisms. Now, all of them can reproduce by fragmentation, hyphal fragmentation or budding in yeast. All of them can reproduce by spores, either sexual or asexual. Now, what about dimorphic fungi? So at the ambient temperature, let's say 73, 74, dimorphic fungi grow as molds. When they get in the human body, though, which is much warmer, right, they start growing as yeast. A lot of pathogenic fungi are dimorphic. One of the examples is here. Histoplasma capsulatum, also known as Ohio Valley fever. It's a fungal infection that is transmitted by inhalation of spores from the environment. The spores usually come from feces of bats or birds. When these feces get aerosolized, people inhale them. As a rule of thumb, people who are immunocompetent rarely have noticeable fungal infections except for the skin or like thrush in the mouth. That makes sense? Anything systemic, no, because they're healthy. But in immunocompromised individuals, this fungus can cause pneumonia, can spread systemically, and if it does, and if it is untreated, it is uniformly lethal. It can be treated. There's a bunch of antifungals. It's not, an inevitable thing. It's not common. As you can see, there's a reason why we call it Ohio Valley fever. It's Midwest. I'm really curious why there's no histoplasma to the west of Rocky Mountain. That is really curious. I don't know. I don't have an answer. By the way, people who are very much at risk are lumberjacks. Right? A lot of bat and bird poo. All those trees fall down, aerosols everywhere, so they are advised to wear PPE. And my second favorite organisms in this course, helminths. They are absolutely adorable. You need to know 3 major taxa, major phyla. Nematodes, trematodes, and cestodes. And you need to understand what's the difference. Nematodes, roundworms with a cylindrically shaped body. They don't have segmentation. They have proper sexes. And they have a pretty developed organ systems with a body cavity. Trematodes, also known as flukes, are flatworms that are not segmented. They are hermaphrodites, and they lack body cavity. Finally, cestodes are the simplest of these worms. They're flat. They are segmented, which I'm going to demonstrate in a minute. Also hermaphrodites, and lack body cavity as well. Effectively, these guys, tapeworms, feed themselves by diffusion of nutrients from the gut of the host. And my favorite of all helminths, I'd say, is a pork tapeworm, Tinea solea. So this is what its life cycle looks like. It's actually quite fascinating. We start with either an egg or a fragment of a tapeworm called proglottid in the soil. When an egg or a proglottid, which is full of eggs, is consumed accidentally by a pig, we're not talking about cows now, by a pig, this egg hatches. The larva and hatches in the intestine. The larva penetrates the intestine and ends up in the pig's muscle. Did you ever buy pig's muscle? It's called pork. Okay? So when this pork is consumed then by a human being, especially, well, not if it is undercooked, the larvae will end up in the intestines of a human, develop into an adult worm, which will use these little hooks on its head to attach itself to the wall of the intestine, and then it's just going to leach a bit of your food. Not a lot. You're not going to lose weight. Most likely, you're not even going to notice. Most people with tapeworm infections are absolutely feel fine. Maybe there's some minor abdominal discomfort. Okay? It tries to parasitize on you as little as possible. Okay? Now that doesn't seem too awful. Like, why are we concerned as much? Well, it turns out if somebody has tapeworm, tapeworms produce a lot of eggs. Hundreds of thousands of eggs a day, actually. These eggs then tossed in feces. As allegedly one of virologists, Robert Schopp, had a plaque on his door saying that the world is covered with 3 inches of feces, meaning that fecal contamination is everywhere. And he was right. So when these hundreds of thousands of eggs come out with poo, when you flush, some of them aerosolize. They end up everywhere, including your hands and so on and so forth. You carry them out in the field, pork, pigs, they eat it, blah, blah, blah. The cycle continues. But what if a human eats such an egg? Human and pigs are pretty close. So if this egg goes into human gut, it hatches, forms larvae, which penetrate the gut, and end up in the muscle, but what's more important, they end up in the brain. This condition is called cysticercosis. If it's a muscle, it's myocysticercosis. If it's a brain, is neurocysticercosis. It is one of the leading causes of epilepsy in the world. Okay? One of my favorite cases was in New York when a family of Orthodox Jews caught neurocysticercosis, which, if you're familiar with Jewish dietary traditions, sounds very suspicious. Turns out it wasn't them who violated Torah, Talmud, whatever. They had a housemaid, which was from Mexico. She was great. But it is known that a lot in Mexico, especially in the rural area, there are villages where all people carry tapeworm. So she did. When doctors were taking samples of the environment, they found eggs tapeworm eggs in the dust on the windowsills. There were so many of them. So it was a question of time. Everybody got treated, everybody got fine, it's treatable. Okay? Cooked pork. All pork that goes into the United States is checked. Obviously, you can't check each and every pork chop. But it is checked. There's monitoring. I can tell you, I kind of worked with USDA when I was in research. They do not mince words. This is one of the most, I don't want to say evil, but they are very picky and very attentive, and they have some laws that can, there are some laws that can put a person in jail for felony if they violate certain agricultural norms. This guy's pretty serious. Okay, and the last bit for today, the viruses, my first, most favorite microorganisms. As we mentioned before, they are acellular, which means they are non-living. Okay? I don't know. There we go. Acellular, non-living. If there will be every question on a board exam, a virus is alive, the answer is no. Good? Most viruses are absolutely benign. There's under 300 viruses that cause human disease known now. They are obligate intracellular parasites. They can't reproduce outside of the cell. They can exist. You can have viruses sitting on that table, but they're not going to reproduce. Viruses completely rely on cellular ribosomes. They don't have them. And they rely on cellular metabolism. They don't have their ways to generate ATP. Structurally, generally, viruses are divided into naked viruses, which consist of the nucleic acid genome and protein capsid, and enveloped viruses like this HIV, which have a nucleic acid genome, a capsid, and the envelope that consists of phospholipids stolen from the cell and viral proteins. This information may sound boring, probably, but not for me, but But it's quite important from the standpoint of microbial control. Turns out, if a virus has phospholipid envelope, it's more sensitive to various chemicals, such as hand sanitizer, alcohol. Naked viruses without that layer are more resistant. I know it sounds counterintuitive, but we'll get to the explanation why. So, for instance, rhinoviruses that cause common cold are naked viruses. The only thing that can prevent the transmission is hand washing. I mean, obviously, not interacting with people also helps, but hand sanitizer doesn't work. That make sense? They were discovered by a Russian scientist in late 19th century, Dmitry Ivanovsky. He called them initially filterable agents because they were passing through the filter that was impermeable for bacteria. So he said that they were filterable infectious agents. First was a plant virus, tobacco, mosaic. They widely range in size, from hepatitis D, which is super small, 20 nanometers, to Pandora virus that can be seen in the light microscope, 700 nanometers. It's pretty huge, okay? So papillomavirus, I chose it not because I don't really like it, like it's not one of my favorite viruses, but it's kind of, hits close to home. So it is a virus. Oh yeah, viruses, since they're not alive, they don't really have a restriction on the type of genome that they have. DNA, sure. RNA, sure. 2 strands, no problem. 1 strand, no problem. So anything goes. So papillomavirus has a double-stranded DNA genome. It's a naked virus, so no envelope. And to my knowledge, it is the most resilient, like resistant to chemicals, among viruses that infect humans. You have to have like pretty concentrated bleach to inactivate this virus. It infects stratified squamous epithelium. I'm not going to torture you asking if you remember ANP1. We're talking skin, stratified squamous, mouth, stratified squamous, reproductive organs, stratified squamous, glans, penis, or vagina, and anus, because it also is covered with a stratified squamous epithelium. When this virus infects that epithelium, it goes into the basal layer. Are you with me so far? The layer in the epithelium that actually divides, because virus needs cellular division to reproduce. It needs cellular enzymes, stuff like that. So it forces cells to divide. When cells divide, there are more cells. Agreed? If it's a skin, this accumulation of cells, we call it a wart. It's benign, can be removed. Okay? Warts can be on the skin, they can be on the anal epithelium, they can be on the vaginal or penile epithelium. So vaginal warts, penile warts, anal warts, no. Okay? But if you force the cell to divide often and to replicate its DNA, this DNA can accumulate mutations. Are you with me? These mutations may lead the cell on the path towards becoming cancerous, which is not good. Cells have mechanisms to kill themselves, just like Jeffrey Epstein did, when they become close to malignant. Okay? So, for some inexplicable reason, some subtypes of human papillomavirus turn off that safety switch, turn off that pathway to apoptosis, to death. And those cells keep going and eventually become malignant. The most known, cancer is not essential. You have to understand that. It is not like, virus doesn't need cancer for anything. By the time somebody develops cancer, it's spread to somebody else. It's done. It's collateral damage. Cervical cancer. Practically all cervical cancer is caused by HPV infection. It also can cause penile cancer, anal cancer, vaginal cancer. It is implicated in certain cancers of head and neck, basically throat cancer, where it kind of works together with Epstein-Barr virus. My point is, we have a pretty good vaccine called Gardasil. One of the best vaccines humans ever made. It is So its structure is such that it is ultimately safe. It can cause, like, obviously, pain at the site of injection, sure. There are no severe side effects at all. It is recommended now for kids before they start active sexual life, like age 11, 12. It is recommended for adults, and I think I just passed the age. I think it's until either 40 or 45, I don't remember. After that, it's like, nah, no, makes no sense. Okay? But I just want to, like, you're going to interact with people professionally. You're going to talk to parents. One of the concerns that people expressed about that vaccine, if you vaccinate a teenager, that then they become just having sex everywhere with everybody. There is no hornier thing than a teenager, period. It does not depend on the vaccine. Vaccine changes nothing. If you think vaccine will make them, lack of vaccine will make them not having sex, you're wrong. So that is not a legitimate concern. And it's extremely effective. And it's been shown that the rates of cervical cancer has been plummeting. Australia has a program to eliminate, eliminate cervical cancer, as well as Sweden, by 2030, sorry. Just now, because the compliance is pretty high. Good? Makes sense? So, thank you for being patient with me. I'm going to, over the weekend, try to work out how can I get more space.